Hello everybody, I'm Professor Steve Forsythe and I'm going to start today a series of 2 videos looking at a 10-year analysis of Cronobacter sakazakii isolates that came from an intensive care unit outbreak. We're going to see the various techniques that have been applied over the years and what they've revealed to us about the organism. A subheading would be lessons on genotyping of Cronobacter sakazakii, a clonal organism. And the aspect of clonality is something I want to bring out in these 2 videos. Almost all the information is essentially in a publication in Current Trends in Microbiology, and you can go on to that paper. There's a link on the various slides here. But I'm going to explain some of the detail which we don't always get to bring out in an academic paper. So the reason why another video on Cronobacter sakazakii is partly because there's still quite a lot of issues coming out in the social media with regard to the American availability of powdered infant formula because of contaminations in a factory with Cronobacter sakazakii. And I've already done 2 videos, one on isolates from the FDA at Michigan, and another one looking at the pathovar of Cronobacter sakazakii, known as CC4. So you can click here or here to get to those particular videos. So this one is going to focus on a broader range of techniques. There's going to be overlap. As usual, I will go off in small tangents. So the structure, we're going to look at the various sources of Cronobacter, which we know about. How Cronobacter gets misidentified, I think, is a very important issue to address in any study. I want to address what is a clonal organism, so you're aware of that. We're then going to go in the beginning, which is an outbreak in neonatal intensive care unit in France in 1994, and how we've applied all these different techniques to those outbreak strains. Where they're going to see the research output and the various background references and who am I. So, as I say, most of the information or links to it and to the various papers can also be found by looking at this one Current Trends in Microbiology article. So, what we're going to look at in part 1, we're going to look at this first box, sources, misidentification, what's clonality. Then we're going to deal with pulse field gel electrophoresis of the strains isolated in France in 1994. So, let's kick off with what is Cronobacter. The Cronobacter genus used to be known as Enterobacter sakazakii. Enterobacter sakazakii no longer exists. It was replaced by a genus. That genus is currently recognized as being made of 7 separate species, of which Cronobacter sakazakii, which is in the news so much, is only one of 7. Now, please bear in mind that with regard to even 16S sequencing, you cannot reliably distinguish between Cronobacter sakazakii and Cronobacter melanoticus. This particular diagram is talking really about the evolutionary history of the species, and our particular interest is in this group here, up with the melanoticus and the sakazakii, because they're the ones which are most associated with human infections. And what's interesting is when we compare genomes, then we notice there are differences in some of the fimbriae, that we don't have the curly fimbriae in melanoticus sakazakii. Instead, we have beta fimbriae. And the other interesting thing when it comes to sakazakii, which is more associated with infant infections, is that we have the loss of malonic acid, but we have a gain in function of using sialic acid. And sialic acid, I think, may be absolutely very, very important in this aspect of the virulence of Cronobacter sakazakii, because sialic acid is found in breast milk, it's found in gut mucin, and it's also found in brain gangliosides. In fact, you can grow Cronobacter sakazakii with sialic acid as a solid carbon source. You can grow Cronobacter sakazakii with gangliosides as a sole carbon source. Because of the association of sialic acid with gangliosides, it is added as an ingredient to infant formula. As I say, it can be used as a growth factor. So, there are, just nothing is an interesting thing on horizontal gene transfer, because there is one little branch in the tertiensis which also has sialic acid utilization, but it's in a different place in the genome. So it seems to be there's horizontal gene transfer going on. Now, Cronobacter, Melanaticus sakazakii in particular, cause infections in adults and infants. The majority of reported cases are in adults, but it's those in infants that hit the headlines and have, understandably, more emotive impact on people. So where do we get Cronobacter from in general? Well, we find it in water. We can find it in animals like mice and chicken. We can find it in insects. We can find it in plant material, salads, herbs and spices, packet soups, cake mixes, and flavored teas. I mean, the favourite of mine when teaching, if people wanted some strains of Cronobacter, then just buy some packets of flavoured teas, and it's great because the little bag is about 2 grams, and you just drop the whole bag into the BPW and isolate your Cronobacter from various steps. So it's a very quick way of getting some Cronobacter, is flavoured tea bags. Just a little tip. Then, if we look at more maybe related to infants, then yes, we can infrequently isolate Cronobacter from powdered infant formula, and we're talking in specifically here for powdered infant formula with a target age less than 6 months, so that's pre-weaning. It's found in water, and water is used to reconstitute the powdered infant formula, and we've known water for reconstitution to be positive for Cronobacter. We find it in breast pumps, and I've already done a YouTube on the isolation of Cronobacter sakazakii and a link to a clinical isolate from breast pumps. People often forget that the type strain of Cronobacter melanaticus was isolated from a breast abscess. And then we do have Cronobacter found in follow-up formulas and weaning foods. These are foods for infants where the infant is older than 6 months, so we're weaning, where the infant formula itself is no longer the sole source of nutrition. And just mentioned that the outbreak in Tennessee 2001, which really highlighted this issue, was actually one of these products and was not, strictly speaking, a powdered infant formula. It's one of those myths concerning the organism. Now, I scooted over nasogastric tubes because it's something I just want to bring out in this particular video for a particular reason. So nasogastric tubes are tubes that go often down through the nose of the baby and they go into the stomach. Why do we do that? Because many of these infections are associated with premature babies, and premature babies are not fed with a bottle but through a nasogastric tube, which is left in place because you distress the baby putting the tube down and bringing the tube back out of the baby. So to depress the amount of stress on the baby, the tubes are left in place. But when you think about it, that tube is going to be at 37 degrees. There can be feed left in that tube, and it's going to be neutral pH, and it's going to be nutritious, plastic surface. What do you think is going to happen? And we've got a series of papers on this with regard to the organisms you find on these feeding tubes. And just find that they have 2 holes at the end so they don't get occluded against the baby's stomach wall. And in those studies, we found bacteria and yeast. These are electron micrographs of used feeding tubes collected from a neonatal intensive care unit. And if we actually take the surface and we use a form of tomography, we can actually visualize the biofilm. So there's the tube wall, and here's the bacterial biofilm on the tube wall, as well as the electron micrographs. The number of organisms was actually totally incredible in these feeding tubes. You're talking about 10 to the 5 to 10 to the 7, which in the UK is about the population size of the city of Nottingham to the city of London. The number of Enterobacteriaceae there was very high, up to 1 million Enterobacteriaceae per tube. And within that did include the neonatal meningitic E. coli, known as E. coli K1. And the paper is here if you want to look more up about E. coli K1 in these feeding tubes. And yes, we did isolate Cronobacter sakazakii from a couple of these tubes when we're doing analysis, and those babies had not been fed infant formula. They were fed the mother's breast milk. So please bear in mind, infant formula is not the only source of the organism. The other issue I want to bring out at the beginning of this video is about misidentifications. Misidentifications may occur through the use of phenotyping. Not always, but often. There's also an issue with regard to if you're using PCR-based serotyping, and I've already covered that in another YouTube video already. So let's just look at some of the phenotyping examples. This we published, oh goodness me, about almost 20 years ago. And this is using two common kits, which were standard in those days, in the ISO standard. These were the kits that were, especially the API 20E was recommended. And these are strains which we can identify according to API 20E. We have 3 strains of Enterobacter sakazakii, as it was known in those days, now known as Cronobacter, not known as Enterobacter sakazakii over here with 32E. Here we have Pentoa, here we have Enterobacter sakazakii, here we have Enterobacter sakazakii, here we have Pentoa. So 2 different kits giving 2 different answers. We followed that up with a later study of a greater number of strains, and with the API 20E we got 82% accuracy and ID32 was 43%. So please be careful using phenotyping. This table just shows you an example of strains which were identified. This is based on whole genome sequencing, so we know exactly what these strains are, and you can see as you go across the different tests, Cronobacter condimenti, Cronobacter maidenski, Cronobacter sakazakii, Enterobacter cloakei, Cronobacter tersiensis, Enterobacter cloakei, and so on and so forth. You can look through the table at your leisure. But unfortunately, some people didn't re-identify their strains when it came to whole genome sequencing and publishing the whole genomes. This is an example from Genome Announcements, and when this paper came out, we had a race in the laboratory as to who could work out what's called the sequence type first. And it says here, complete genome sequence of Cronobacter sakazakii strain CMCC 45402. It took 15 seconds to find out this was misidentified species, because we could just quickly, within 15 seconds, I looked up a key allele, which is known as fuse A allele in the MLST scheme, and it was the wrong allele for any Cronobacter sakazakii that we knew of. It was exactly the allele which was always found in Cronobacter melanaticus. This strain is Cronobacter melanaticus. We know from the whole genome it's Cronobacter melanaticus, and despite contacting the authors more than once, despite contacting the journal editors more than once, never been any response to this issue. So one has to be very careful not to always believe absolutely everything, but to take the opportunity to double check. So let's move on to what's a clonal organism. We're taught Microbiology 101. Bacteria go 1, 2, 4, 8. And when you just take that on face value, it means that those 8 isolates, those 8 bacterial cells, will be clones. That generally, that DNA sequence of each cell will be identical to the first one. That's what clones are. They are indistinguishable. But then we're taught in Microbiology 101+1 about mutation and transformation and transduction. And so we know that when a bacterial cell divides, as DNA replicates, then mutations can occur, we get variation. And therefore, you would get DNA that will be differing between strains because that DNA change has not been corrected. So therefore, you can use techniques to distinguish the strains, which is what is commonly portrayed in textbooks. Textbooks give a straightforward story most often. In actual fact, there are organisms, such as Cronobacter sakazakii and Salmonella enteritidis, not Typhimurium, but Salmonella enteritidis, which are clonal, where the recombination events occur such that it may only recombine with its own sequence type, or it's corrected in some way. And so what happens is that you're going to get the same pulse field bar pattern, but those strains are unrelated. They're indistinguishable, but what's happened is maybe the DNA variation has been corrected very accurately, or it's not been allowed. And so what happens that you get this division of strains, ends up in different countries, different decades, nearly indistinguishable. But they're not the same. They are unrelated. Now, as I say, the main focus of this is going to be looking at a set of strains that have been analysed over 10 years. The first paper is this one from 2007, and we're going to look at the results that were published over a 10-year period coming up to 2017. So we're going to kick off with this paper back in 2007. So here it is, genotypic and phenotypic analysis of Enterobacter sakazakii strains from an outbreak resulting in fatalities, sadly, in the neonatal intensive care unit in France. And huge thanks to Marie-Francois, who had the foresight to put the isolates into the freezer when they were first found, and then contacted us when she felt the techniques were now available to reanalyze those strains. Now, disclaimer, just for ease, I'm just going to disclaim completely now that although in 2007 these strains were called Enterobacter sakazakii, they were later identified by us as Cronobacter sakazakii. For continuity, as I go through these slides, I'm going to keep saying Cronobacter sakazakii, even though the original paper said Enterobacter. This absolutely does not mean all E. sakazakii equal C. sakazakii. That's not true, that's not accurate. Enterobacter sakazakii is a former name for the Cronobacter genus. The Cronobacter genus is made up of 7 species, of which 1 is Cronobacter sakazakii. So I will say Cronobacter sakazakii, and that's because that's what those strains were later identified as. So here's the list of strains. We have neonates, obviously anonymized, they've got letters. You can see that 4 neonates died. You can see the sex of the neonates. You can see the mode of delivery. We have vaginal and we have cesarean. So where sometimes I've known a mother being told that she infected her baby with Cronobacter through the vagina, but here we have cases that with cesarean as well as vaginal. We have gestation age, so you can see the prematurity of them, the birth weight of them, when they started to feed, and the onset of symptoms after they started to feed, and we have what their symptoms were. NEC means necrotizing enterocolitis, which is where the intestinal wall dies, it degrades, and therefore the great worry is that the bacteria in the intestinal contents can therefore cause a systemic infection. So it's a very, very serious infection. We have septicemia, which is bacteria growing in the blood, but you can see we have meningitis here as well. But we also have some asymptomatic, digestive problems, asymptomatic, and we have sepsis. So we have a variety of symptoms here. Very interesting strain collection from Marie-Francois. When we did, in those days, the technique that everybody used, which was pulse field electrophoresis, we found there were 4 clusters of strains. So there wasn't one, in a sense, strain causing all these infections. There were 4, to the best of our knowledge in those days, 4 strains causing the outbreak. So if we now reorganize those strains according to the pulse type, And after the pulse type, we then organize them according to date of isolation. Then we start to see some interesting stories coming out. The pulse type 1 will be the first group of strains, and the first infection were in March through to June. We have 5 babies here. The first one we have no details, but it's the first one. These weren't kept. We have 2 necrotizing enterocolitis. We have 2 asymptomatics. See, some isolates were from the trachea, some from the stools. The second pulse type 2, greater number of isolates, and you can see that we have 3 deaths. You can also see that 2 strains have got ESBLs. So there's a big difference here. Deaths, ESBLs going on. You can see again we got perinatal fluid, sputum, trachea, stools, conjunctivitis, skin, stools coming down, and we got a lot of necrotizing enterocolitis, and we have meningitis as well. Then we have the third pulse type, digestive problems, asymptomatic. So we go from a group where we're getting deaths and meningitis to a group where we're getting digestive problems or asymptomatic. And those 3 isolates are not, they're not from a patient, they're actually end of bottle or they're prepared feed. And so they're indistinguishable strains from the stools of these 2 babies, from the isolates from the end of bottles and from the prepared feed as well. Then, interestingly, if you look carefully, you'll see the letter B coming up 4 times. We actually have 4 isolates from the same baby. And you can see the dates of them. So you can see this baby was colonized or infected by the baby over a couple of months, and actually it had a different strain earlier. So this baby shows that babies can be infected by more than 1 strain of Cronobacter, and that's very important take-home point with regard to how many colonies you pick off and identify, etc. Then we have a 4th group, and these 3 strains came from infant formula on the same day, and they're all on the 11th of July. And okay, that looks like a smoking gun, but when you look at it, they're July, and virtually all the other isolates were before then. They only looked at the infant formula after all these cases had occurred. Then we got this baby at the bottom, and the reason why the baby, that particular one is out of the chronological order, is because when it came to looking at the baby isolate through 16S, as we did in those days, then actually it turned out to be a E. cloacae. It has been misidentified. It's one of those examples where phenotyping had misidentified the strain, and it wasn't Cronobacter sakazakii, it was Enterobacter cloacae. Unfortunately, that baby did die from sepsis, and it occurred at the same time, roughly, as the other babies were infected. So what we've got is, in this outbreak, and this is just part 1, is that we have the issue of misidentification occurring through phenotyping. We found that by using pulse field gel electrophoresis that we were able to find that this so-called outbreak was actually composed of 4 pulse types of Cronobacter sakazakii, and in actual fact there were 3 pulse types concerned with clinical cases, so there were 3 overlapping outbreaks, and it shows that babies can be infected by more than 1 strain of Cronobacter sakazakii. 2 pulse types were isolated from prepared feed and end of bottle. 1 particular pulse type was associated with 3 deaths, and that gave us, and I had to very cautiously, I put a statement in that paper back in 2007 saying possibly these results indicate that some strains of Cronobacter sakazakii may be more virulent than others. just based upon this one outbreak. And we're going to see what a like a prophecy almost that was with regard to the future when we come to part 2. So the other interesting thing to bear in mind, how were the babies fed? And I said before about nasogastric tubes, and that's really important to look at how babies are fed when you have an outbreak situation, because what happened in the situation in France is that prepared formula was made in a large volume, not per feed, but was made up in a large volume, put into a big syringe, and this syringe has a pump mechanism, and slowly the syringe plunger is pushed down, pushing the feed along the nasogastric tube into the baby. That pump was at room temperature, which in a neonatal intensive care unit is warmer than normal ambient temperature. So plenty of opportunity for potential bacteria to grow, as well as attaching to the plastic. So moving on, what we're going to be looking at in part 2, we're going to be looking at the number of papers that were subsequently published concerning these strains. We're going to look at the outputs and research and our knowledge of Cronobacter sakazakii based upon these strains, and we're going to be looking at a range of techniques of genotyping, ranging the pulse field gel electrophoresis. We just looked at different types of MLST, CRISPR-Cas array profiling, and SNP analysis. And the bottom line literally is indistinguishable, does not mean the same. So just going to finish this part 1 with regard to showing you some useful references. By all means, please email me if you want any assistance. If you want to just find the list of publications, they're here on this site. And I hope you enjoy this, and I hope you tune in, so to speak, for part 2, where we're going to be greatly expanding the story. And the last one is simply telling you who I am, if it's of interest to you. So-called retired doesn't mean that much. Advisor to WHO on the safe feeding of infants, and written lots of papers, and primary research, Cronobacter and infant formula. I'm curator of the Cronobacter Pub MLST database. So thanks very much. I hope you enjoy this and that you are able to tune in and see part 2 as well. Thanks very much.